Now we continue the webinar. It's a pleasure for me to introduce a colleague, a friend, a very good friend, Professor Luigi Boccuto from Clemson University of South Carolina, USA. And the topic of his speech is genetic factors in NAFLD. Please, Luigi, I remember you that you have 20 minutes, please.
Yes. Yes, well, thank you very much for that kind introduction. It's a pleasure for me to be here. This is a very interesting topic, and it's a very complex one. It's complex because the phenotype, the clinical manifestation of this condition is extremely variegated. And you can see here already from the cover slide how we deal with different stages of this disorder. But as we will see soon enough, the complexity goes on with the clinical characterization and diagnosis of this condition. I will not spend too much time on the clinical features because other more renowned and more expert speakers will cover that. But I just want to focus on the pathogenicity and the mechanisms that are underlying the onset and then progression of non-alcoholic fatty liver disease. As you can see, the clinical definition is based, of course, on the features that can be assessed. So we're talking about clinical assessment, imaging, laboratory tests, and of course, biopsy. But we do have different categories that are interrelated. So there is a progressive dimension that we need to consider. There is increased severity of the manifestation, and there is an increased decline of the and degradation of the liver functions. If we focus on the different subgroups of NAFLD, we can see that this complexity is even exacerbated by the presence of comorbidities. In fact, the alcoholic fatty liver disease is often present as well, but also other conditions that really open the doors to different pathogenic mechanisms involving metabolic disruption, immune imbalance, toxicity from diet and other environmental factors. So if we want to have a really encompassing approach to the characterization of the causative mechanisms underlying this condition, we must take into consideration the complex nature of this disorder. And if we focus on the dynamics of the onset of this disorder, we can see that they are closely interrelated with other organs. In fact, if we narrow down the pathogenicity of NAFLD, we can see that there are influences from organs around it. So first of all, the effect of the diet, but the interaction with the adipose tissue, the pancreas, the intestine, as was mentioned before, the leaky guts, and all the interaction between bacteria, the immune system, and then the ultimate damage on the hepatocytes. But that's not the only direction of this interaction, because there is an interaction from the liver to the other organs. And that is important because we have to contextualize NAFLD in the wider spectrum of the metabolic disorder. As we saw before, there is a series of comorbidities of disorders that can be detected in individuals suffering with NAFLD. And this is a summary of the possible pathogenic interactions where we have disruption of the lipid metabolism starting from the liver and then spreading towards mostly two main mechanisms: the generation of insulin resistance that starts in the liver and then spreads to the other organs, and the induction of inflammation. The critical point about the inflammation is the chronic feature of it. So it's not just an acute event, but it's something that once is established, it remains and affects not only liver tissue, but several other organs. So this is quite important to understand the systemic consequences of NAFLD. And since we're talking about a complex disorder, it's good to take a step back before we approach the genetic factors involved in these complex disorders. The definition of a complex disease is the interaction of environmental inputs and the metabolic response based on the molecular and genetic predisposition of each individual. The characteristics in terms of pathology of these disorders are that they do not have a Mendelian mode of inheritance. So even if there are genetic features that may be very relevant, as we will see in a couple of slides, the segregation in the families, the pattern of inheritance, the distribution in the population, they do not follow the Mendelian pattern, which makes it more complicated for systemic assessment during, for example, counseling for the family or large population screening. The other point is that even if there is a genetic component, is quite often not monogenic. So there are several genes involved that contribute to the overall phenotype. And sometimes the quantitative assessment of the contribution of each gene is quite complicated because the threshold determined in the for the interpretation of the pathogenic effects of genetic variants are designed to assess the contribution of single variants. But in this case, we might have a network of multiple genes, maybe even multiple pathways that concur to determine the onset and progression of the liver disease. And then the third element of complex disease is the environmental factors. The environmental factors may play a role that may vary from simple trigger when the genetic component is relatively strong to the sole causative effect. So again, it's very complicated to assess what is the actual contribution of these environmental factors because they may vary from individual to individual. So once we have an overview of a complex disease, particularly related to NAFLD, what is the role of genetics? And what is the role of genetics now in the post-genomic era? Researchers consider the post-genomic era this period of time after the final results of the Human Genome Project have been shared, after the next generation sequencing, the large sequencing approaches have become more available in terms of cost, in terms of logistics. And so they open the doors to the world of omics, where the omics are really the ones that are targeting the DNA, so the genomics, as you can see here, the RNA, the transcriptomics, that started with the messenger RNA, but really now is extended to all types of RNA, proteomics, proteins, interactomics, so the interaction of multiple proteins, and metabolomics. So as you can see here from this cartoon, these approaches are aiming to assess at the vast level, the very broad level, the possible alterations in our pathways, in our metabolism, and they are targeting the DNA, the RNA, proteins, and the interaction of proteins, and then finally the metabolomics with all the metabolites, so the product of these proteins. So this will give us really a very encompassing approach that will help us understanding the link between genetic factors and the final clinical presentation. And this is possible through the functional genomics. So an approach that is focused on isolating individual alterations or single alterations in the gene sequencing or non-coding sequencing, and then through high throughput technologies, extending this analysis, this investigation to the whole genome, collecting data and interpreting data through bioinformatic analysis. So really introducing that multi-layer dimension where we can connect different alterations and then analyzing, interpreting the results. And I would like to add another step to this graphic, which will be the translational step. So applying the information collected from this functional genomic approach to the clinical practice. So really bringing the information from the lab to the bedside of the patient. And so in order to do this, today I want to speak about the connection between genes and liver. And I like to use an example to summarize three main categories. We can use an example for genes where we can have the good, that is HSD17B13, the bad, PNPLA3, and the ugly, which is CIP2D6. Why did I pick these? Of course, there is a citation, the famous Italian movie, but we will see how these genes really represent a paradigm of different potential effects of gene variation on the liver metabolism. We start with the good, which is this gene 17 beta hydroxysteroid dehydrogenase 13 that encodes a liver-specific enzyme that targets steroids, pro-inflammatory lipid mediators, and retinol. So if you remember, when we had an overview about the pathogenic mechanisms underlying NAFLD, one of the critical steps was the chronic inflammation. So this is an enzyme that really works by expanding, amplifying, exacerbating the immune response and promoting inflammation at multiple levels. So loss of function variants, so inactivating variants in this gene, have been associated with protective effects against NAFLD. So that is why we label that the good. And this is important not only to stratify the general population for their overall risk of developing NAFLD, but also because it provides potential molecular targets for treatment. And this is the translational dimension we were mentioning before. So variants in this gene may prevent or delay the progression of NAFLD. So if you remember the steps from steatosis to non-alcoholic steatohepatitis, and then liver fibrosis, cirrhosis, and hepatocellular carcinoma, all that progression at every step of this progression may be either prevented or delayed, depending on the overall effect of these variants, by HSD17B13 variants. Even, and this is a recent study, even in individuals that are carrying the variants on the bad gene, the PNPLA3. So you can see how there are many actors at play here, many potential genetic factors playing, and some of them are playing against each other. So that is why the multi-gene network is important, because we need to assess the overall contribution of genetic factors. So overall, what we learn is that inhibiting the activity of this protein through RNA interference and small molecule agents may prevent NAFLD progression. Then we move to the bad, PNPLA3. This is a gene that encodes a patatin-like phospholipase domain containing 3 protein, also called adiponutrin, that is a very important intracellular membrane protein that is triggered after feeding and during insulin resistance and promotes lipogenesis. So remember, again, if we go back, if we look back at the pathogenic mechanisms that promote onset and especially progression of this disease, insulin resistance was a very important step. Lipogenesis, so disruption or dysregulation of lipid accumulation is another important step. And the very interesting thing about this gene is that there is one variant, this isoleucine to methionine missense change affecting the amino acid residue 148, that not only is prominent predisposing genetic factor for non-alcoholic fatty liver disease, is also a prominent predisposing factor for alcoholic liver disease. And to my knowledge today, is the only genetic variant that is associated to both alcoholic and non-alcoholic forms at very high level. But it's relatively frequent in several populations. You can see that the estimate on the overall population is around 20–26%, but it may vary in different ethnicities. And in the highest points in Latin American ethnicities, it may even affect or may interest more than half of the population. So this is a very important issue for community medicine, because stratification of the risk in individuals from different ethnicities may be extremely important for the management and treatment of NAFLD. So here we can see summarized the effects of this variant, where the wild type isoleucine amino acid in PNPLA3 148 position is responsible for a balanced transition of lipids through the membrane, and so it keeps the lipid droplets under a certain size. And instead, if we have the methionine, there is a promoted lipogenesis with the generation of macrovesicular steatosis. So there are these big vacuoles, this big accumulation of lipids in the hepatocytes. And then we move to the ugly. Now, why did I label this the ugly? Because it really creates a lot of damage, even if it doesn't seem to have a clear purpose. So like the character in the movie that is sort of messing around, going one direction or another, this one is not directly involved in NAFLD, but I wanted to pinpoint the role of this gene because it's extremely important in pharmacogenomics. And for a complex and systemic disorder such as NAFLD, the evaluation of the pharmacogenomic predisposition, that the genetic background underlying interaction with drugs is critical, because as this condition affects multiple systems and multiple organs, therefore the management and treatment must take into account medicines, pharmaceutical compounds that will target multiple organs. And this is a gene that encodes a monooxygenase that localizes in the endoplasmic reticulum of hepatocytes and metabolizes, just this protein metabolizes about 1/4, about 25% of the commonly prescribed drugs. So it's part, of course, of the P450 family, the largest cytochrome family in the liver, but it's responsible for the way our body responds, interacts with 1/4 of the commonly prescribed drugs. And you can see here there are drugs from different categories and different families, from antidepressants to antiarrhythmics and analgesics. The thing is, this is an extremely variable gene in humans. It's highly polymorphic. There are multiple alleles recognized for their overall effect, and defined as poor metabolizer alleles, rapid metabolizer, or ultra rapid metabolizer when you have 3 or more active copies of the genes due to the gene duplications. So again, this is not a specific NAFLD gene, but when we consider the genetic impact on the onset, progression, and management of complex disease like NAFLD, we cannot forget to mention this gene and its pharmacogenomic role. But these were just examples. In reality, the interaction between genetic factors and environmental factors is way more complex. And we have genes like the PNPLA3 that have a specific and straightforward role in promoting lipogenesis and chronic progression. We have genes that have a more articulated role in interacting with environmental factors promoting oxidation. So they promote the oxidation at the mitochondrial level. And we have, of course, genes and epigenetic factors, as the microRNA examples listed here, that interact with the immune system. So interact with the Kupffer cells in the liver, but also with leukocytes at the systemic level. But all these, as you can see, is really intertwined and it's interconnected because one thing induces the other and vice versa. So the complexity level of the pathogenesis of NAFLD really forces us to consider all the genetic networks, all the pathways that are involved, and how individual contributions play in the whole equation with all the others. And just to give you an idea, we mentioned, of course, so far genetic factors, but we forget about epigenetic factors. The microRNAs are just some of those, but they have been clearly associated with the progression from step to step of liver disease from the normal level to the first stage liver injury, hepatitis, and then fibrosis and cancer. And then just to finish, we must not forget that the genes are also showing us the way for developing new treatments. So this is the translational dimension we were mentioning before, and here is just an example of how drugs may target different nodes of the pathogenic pathway leading to liver disease. At the level of the oxidative stress for antioxidants, at the level of growth factors and other ones on acting on the immune system. So in conclusion, we're living now in an era where we have an increased availability of omic technologies. And the data that have been collected and are currently collected are really suggesting that the implementation of genomic profiling of subjects affected with NAFLD, or even better, at high risk. So we can introduce a level of management at the preclinical stage, are really critical, and they can critically improve the outcome of this disorder. Therefore, we want to apply these to the precision medicine protocols. And the benefit of that will be developing screening protocols for subjects at risk. For example, identifying individuals carrying the PI148M variant in PNPLA3, characterizing the potential biomarkers for the preclinical selection of ideal candidates for specific treatments, and even determining the optimal dosage of these treatments. For example, looking at the variants in CYP2D6 and their pharmacogenomic effects. And another potential benefit is identifying molecular targets for the development of new treatments. For example, inhibiting the HSD17B13 activity through RNA interference. So as you can see, the genetic factors are playing a very important role in the pathogenesis of NAFLD, but they're also providing us with very powerful tools for the management of this disorder. And with this, I'd like to thank you very much for your attention.